The foundation of culture-based testing relies on growing and isolating microbes in culture before conducting follow-up diagnostics. This means that it is slower and less sensitive than modern molecular and genomics approaches, but it is by no means obsolete. Culture-based testing remains the gold standard to confirm the identity of specific types of microorganisms, and we will go through the process from first principles. We will discuss the use of selective and differential media, antibiotic susceptibility, and biochemical or immunological testing in culture-based analyses. The body harbors a vast number of bacterial species as normal flora, and we will demonstrate how culture-based testing can differentiate between Staphylococcus and Streptococcus species as clinically significant examples. Staphylococcus species are gram-positive cocci, usually found in grape-like clusters. They can be easily confused with Streptococcus species, which are found in pairs or linear chains of gram-positive cocci when looking down the microscope. To tell them apart, we perform the Coagulase test. Take a small part of a suspected colony and place it on a glass slide while adding a few drops of hydrogen peroxide. A positive test is one where gas bubbles appear. Staphylococcus should be Coagulase positive, while Streptococcus is Coagulase negative. Within Staphylococci, Staphylococcus aureus is the species of greatest medical importance as it is commonly associated with infections in hospitals and nurseries. Staph epidermidis and Staph saprophyticus are the other clinically significant staphs. Staph epidermidis has been associated with bacteremia, endocarditis, wound infections, while Staph saprophyticus is a common cause of urinary tract infections. We have a number of culture-based tools at our disposal to tell these staphs apart. Selective media selects for the growth of one specific type of bacteria, while inhibiting the growth of others. Differential media targets the properties of one type of bacteria, which leads to a change in color when the targeted bacteria is present. When combined, selective and differential media create a useful set of conditions to identify microbes using culture-based methods. Blood agar is a non-selective, general-purpose medium that supports the growth of most bacterial pathogens. The presence of blood provides some form of differentiation, as specific microbes can lyse the blood and lead to hemolytic zones of clearing. Typical colonies of Staph aureus on blood agar are yellow and often hemolytic. Mannitol salt agar has a high salt concentration, which selects for Staph aureus by producing colonies surrounded by a bright yellow zone. Other Staph species are surrounded by a reddish-purple zone on this agar. Staph aureus is the only Staph species that contains the enzyme Coagulase, and we can run an immunological test to detect its presence. We rub a colony of our suspected microbe into the Coagulase reagent as well as the control. If Coagulase is present, the reagent containing antibodies against Coagulase will clump the bacterial cells together in what we refer to as agglutination. This will look very different to the control reaction. But what about the other Staph species? How do we tell Staph epidermidis apart from Staph saprophyticus? They can be differentiated based on sensitivity to the Novobiocin antibiotic. We place a Novobiocin disc onto an agar plate that has been inoculated with the unknown staph. If Novobiocin is inhibiting its growth, it is Staph epidermidis. If not, it is Staph saprophyticus. Streptococci are gram-positive cocci, characteristically arranged in pairs or chains. Their ability to hemolyze red blood cells to various degrees, growth on selective and differential Mitis Salivarius media, as well as susceptibility to bacitracin and optochin antibiotics are all important criteria for their classification. Hemolysis on blood agar is the first point of differentiation across streptococcal species. Alpha hemolysis is characterized by an indistinct zone of partial destruction of blood cells around the colony, often with a greenish to brownish discoloration. Beta hemolysis is a clear colorless zone around the colonies where the blood cells have undergone complete discoloration. Strep pyogenes and Strep agalactiae demonstrate beta hemolysis, while Strep mitis and Strep pneumoniae demonstrate alpha hemolysis. Mitis Salivarius agar is a selective and differential medium that is inhibitory to most coliforms and selective for Streptococci. Strep pyogenes, Strep agalactiae, and Strep mitis all form small blue colonies about 0.2 mm in diameter. Strep Salivarius produces larger blue colonies 2 to 5 mm in diameter and with a raised gumdrop appearance. Antibiotic susceptibility is another key differentiating factor amongst Streptococci. Bacitracin can be used to distinguish Strep pyogenes, sensitive, from Strep agalactiae, resistant. Optochin can be used to distinguish between Strep mitis, sensitive, and Strep pneumoniae, resistant. We can also use a commercial immunological testing kit to differentiate Streptococci by their Lancefield Antigen. Each reagent contains an antibody targeting different surface antigens unique to each type of strep, and we are looking for positive reactions in the form of agglutination. The strep should only react with one of these reagents, and in this case, our sample reacts with group A antigen, confirmation that it is Strep pyogenes, also known as group A strep. Culture-based diagnostics involves a cascading series of tests, the results of which leads us to increasingly specific criteria for microbial identification. You should now be able to describe the value of selective and differential media, antibiotic susceptibility, and biochemical or immunological testing in culture-based diagnostics.